Welcome to the first episode of TakeNote. Today, we're going to be covering the topic of DNA replication. DNA replication is how cells duplicate their DNA. It's usually done before cell division to ensure that the daughter cell has virtually identical DNA to the parent cell. DNA replication in eukaryotes is split into three parts: initiation, elongation, and termination. At the start of initiation, the protein helicase begins to unwind the DNA binding to a site called the origin of replication. From this site, helicase begins to split the two DNA strands, kind of like a zipper. Then, proteins called single-stranded binding proteins bind to the DNA to hold it in place and keep it from annealing back together. During elongation, the next step, a protein called primase attaches RNA primers to both DNA strands while the DNA is still being unwinded by helicase. The two strands of DNA separated by helicase are given two names: the leading strand and the lagging strand. The leading strand is the DNA being unwound from the 3' to the 5' direction, and it only needs one RNA primer to be added to it. The lagging strand is the strand of DNA that is being unwound from the 5' to the 3' direction. For the lagging strand, multiple RNA primers need to be added every few couple of base pairs. These primers are important in the next step, where DNA polymerase III binds to the primers and begins adding DNA nucleotides in the 5' to 3' direction. On the leading strand, DNA polymerase III is able to continuously add DNA nucleotides without stopping. However, on the lagging strand, DNA polymerase III must start at one primer, move in the 5' to 3' direction adding nucleotides, and stop at the next primer. This starting and stopping on the lagging strand is due to the fact that DNA polymerase III cannot add DNA in the 3' to 5' direction. Each of these small fragments of DNA that have now been created on the lagging strand are called Okazaki fragments. Once the process of elongation is complete, the process of termination begins. DNA polymerase I binds to the RNA primers, removes them, and attaches DNA nucleotides to take their place. This happens on both strands of the DNA. Between each Okazaki fragment and newly added DNA nucleotides to replace primers, there is a little bit of gap. This gap is filled in by a protein called ligase. Ligase forms a bond between the two DNA nucleotides that have a gap in between them. After the replication process is over, the one strand of DNA that we started out with has now formed into two. Each strand has one strand of parent DNA and one strand of daughter DNA to form a double helix. Wow, looks like we've covered the entire process of DNA replication. I hope you all had a great day and you now understand this topic. Hope to see you next time on TakeNote.